Hello and welcome to this e-seminar, which will provide an introduction to databases for non-targeted food authentication analysis. This e-seminar will provide the listener with a general introduction to the subject of databases for use with non-targeted multi-analyte analysis methods. Topics covered in this short presentation will include a brief introduction to targeted and non-targeted databases, fundamental steps in database construction, as well as aspects of database validation and curation. It has been estimated that approximately 22% of the global food supply may be the subject of food fraud. Most of the current official testing methods employed are only capable of identifying the presence or absence of known ingredients or adulterants. Testing for adulteration is difficult due to the variety of substances now used to adulterate foods and is reliant on surveillance intelligence to indicate which adulterants may have been employed. Recent technical advances are leading to the development of non-targeted multi-analyte methods of analysis which address this problem through comparison of sample molecular fingerprints with those generated for reference samples. Fingerprinting reference datasets are generally maintained in specialized databases. This e-seminar will introduce the viewer to some of the key aspects of these databases and discuss how they are constructed and the aims and objectives they are designed to address. The table presented on the slide provides an indication of the diversity of compounds that have been identified as being used to adulterate foods for the purposes of economic gain. The content of the table clearly illustrates the problem faced by analysts in that it is not practical to test all food products for each potential adulterant listed. This is where the advantages of employing non-targeted multi-analyte analysis excels, in that the analyst does not need to test for each individual adulterant, but needs only to compare the molecular fingerprint of a test sample to that of an appropriate reference, in order to ascertain if an ingredient compositional discrepancy exists. Those analytical methods currently employed for food authentication purposes may be subdivided into two types. Type 1 consists of the targeted analysis of specific analytes or marker compounds naturally or not naturally occurring in the foodstuffs that may be characteristics of a particular adulterant, e.g. melamine and other compounds, or characteristic of a particular foodstuff, e.g. concentration of methyl glyoxal for specific active manuka honeys. A database is not required with this approach as the target analyte is either present or absent. Type 2 consists of non-targeted fingerprinting techniques. These simultaneously measure a range of analytes or markers and then compare them to established reference data, e.g. 1H NMR analysis of honey for global point of origin determination. A database is required with this form of analytical approach in order to know what analytes are naturally present in a sample and at what concentration. An example of the type of database required would be that for stable isotope ratio analysis. Databases employed with targeted analysis are the most established in food. In general, targeted analytical methods are comparatively easier to transfer between laboratories than untargeted methods. Critical to comparability of targeted analysis results is use of robust and validated analytical methods, laboratory competence in performing the analysis, verification through participation in appropriate proficiency testing schemes. An example of an established targeted analytical database for foods is that of the Food Allergy Research and Resource Programme Database, or FARRP, which is maintained at the University of Nebraska, Lincoln in the USA. Databases for use with non-targeted analytical methods are comprised of organised collections of reference datasets. Of critical importance is that the datasets should be generated using established, validated analytical protocols, where possible from certified reference materials, and compiled from representative numbers of authentic samples. Inclusion of test samples, even when an analysis indicates that they are authentic, should be avoided. Only samples of known provenance should be employed. This is because the use of datasets is critical in defining the natural variability of a property of a foodstuff or an ingredient. This natural variability is used as a reference for comparison when analysing test samples. When test samples exhibit a marked deviation from the expected degree of variation, then this is frequently taken to be indicative of food fraud. The incorporation of misclassified samples into the database may incidentally result in the introduction of bias and the generation of incorrect query results. In terms of content, non-targeted analysis databases are generally comprised of data derived from spectroscopic or spectrometric fingerprinting-based analyses, which consist of collections of spectra or spectrographic data. No specific analytes tend to be included for the samples employed with database construction, but application of the data requires the use of robust statistical analysis followed by reporting of any relevant findings. A major disadvantage of this format of database is that the data generated may not easily be employed for different authenticity problems, and so tend to be restricted in the potential applications they can be employed with. There are 7 fundamental steps employed in database construction and which are listed on the slide. The first step involves defining the purpose of the database, including intended function, expected outputs, and the listing of associated parameters such as the scope and analytical methods employed. The second step is reviewing the types of information that will need to be recorded, such as ingredient name and source. Step 3 encompasses the organisation of gathered information into primary and secondary items. For example, ingredients and ingredient sources. The defining factor here is that each primary item becomes a table. During step 4, the data generated by the analytical methods is allocated into the primary item tables defined during the information organisation stage. In step 5, a primary key is specified, which is a column or a set of columns which can uniquely identify a row. In step 6, the relationship between different tables is defined, and the analytical approach or algorithm to be employed for query processing is selected. And finally, the ability of the database to return the appropriate response is tested, evaluated, and refined where appropriate. The first critical step is to define the purpose for which the database is intended, and then list all the associated parameters. In terms of parameters, these may include the scope of the database, methods employed for data acquisition, approaches for sample preparation if required, identification of appropriate reference materials, how to evaluate database performance and validation, considerations for database storage, backup, and external access, the matter of curation to ensure database contents remain up to date and valid. This list is not exhaustive, but most of the key points will be covered in more detail in the following slides. The process of defining the scope of a database is the first critical construction step and establishes the key attributes of the final database. Key considerations will include who will be the end user and what will be the intended purpose of the database. For example, where the food business operator is the end user, then the purpose will generally be for due diligence testing and to act as a deterrent to fraud in the supply chain. Defining the purpose of the database will then determine its potential applicability. What analytical method or methods will be employed? What samples will need to be analysed? Which reference samples will be needed to define the scope of the database, taking into account that the samples used to populate the database need to be comparable to those that it will be challenged by. The choice of analytical method is an important consideration and will depend on the physical and chemical properties that differentiate a product. Examples of typical non-targeted multi-analyte analytical methods include near-infrared spectroscopy, or NIR, Fourier transform infrared spectroscopy, or FTIR, Raman spectroscopy, nuclear magnetic resonance spectroscopy, or NMR. Methods such as GC or LC mass spectrometry have been reported to be unsuitable owing to the variation introduced to the results from differences in sample separation columns, etc. The selection and acquisition of appropriate reference samples is also critical to the utility of the database. Data for reference samples within the database need to be both authentic and relevant. The use of synthetic admixtures should be avoided, as the impact of how they were generated may mask that resulting from any adulteration. Inclusion of a fraudulent sample labelled as an authentic sample may potentially invalidate the database. Precautions need to be taken in order to ensure that authentic reference samples differ between commodities and should ideally be a certified reference material if available. If not, it should be collected from primary producers, i.e. farms, fisheries, etc. And ideally, it should be collected by impartial collectors. With non-targeted analytical approaches, the sample population should ideally reflect the natural variation observed for the analyte or analytes of interest in the target population. In many situations, it may be necessary to consider any natural variation that may be caused by a variety of factors, such as geographical location, climate, variety, or even breed. In addition, the multivariate analysis techniques frequently employed may also require a sampling size that is sufficient to build a fingerprint that is representative of any authentic samples. As well as these considerations, the final sample size may also depend on a range of additional factors such as access to authentic samples, project budget, working time frame, and the logistics of sample collection. The collection of analytical data is a critical step in the construction of any database, but in the case of those for non-targeted multi-analyte analysis, additional contributing factors may need to be considered. Therefore, the process of data collection should include all possible information relevant to the purpose of the database. To assist with this aspect of database construction, guidance and best practice on the minimal reporting requirements that should be adopted has been published by the Chemical Analysis Working Group for Metabolomics Standards Initiative, the reference for which is listed on the slide. As highlighted, analytical data collection requires consideration of factors that may not be directly related to the sample analysis, but that may influence the results and should be controlled. Examples of extraneous influences that should be considered include change in ambient laboratory temperature, instrument variability, minor changes in sample preparation procedures. Effective strategies for control may include a combination of monitoring of known or suspected influences, analysis of reference sample quality during database creation, random order collection of analytical data. Basic database structure comprises of two components. Firstly, the software underlying the database structure. This controls the physical storage of the data, organises the data into tables, and establishes the links between different tables. The second component is referred to as the application layer and provides the interface between the database and the user. It streamlines the process of working with the database, controls data filtering and sorting requests generated by user queries, and regulates additional functional aspects, including administrator and user access, e.g. password protection, data input and export. There are several factors to consider when designing or choosing a database structure. These may include what has successfully been used previously with similar databases, for example, MySQL, Oracle, MongoDB, etc. Is it freely available? How secure is it? What volume of data is it rated to? What are the stated speeds of data entry and retrieval? Does it have inbuilt backup capability, or can it be added? Can it be modified in order to accommodate updates, i.e. new metadata? Applying databases for non-targeted testing approaches requires the identification of authentic sample profiles or fingerprints. Profiles generated using multivariate models has been reported to be preferable, as may permit the differentiation of authentic to non-authentic with greater certainty. The application of a database to the analysis is generally a two-step process. Firstly, some form of data compression is undertaken both to reduce the size and complexity of the dataset. Modelling is then undertaken using selected features. The modelling approaches generally employed may be broadly classified into two categories. These are either discriminant analysis, which is appropriate where determination of a value is required, such as the oxidation level of an ingredient, or class modelling, which can be either defined as a single class, for example if a sample is authentic or not, or multiple classes, for example if olive oil is of Italian, Spanish or Greek origin. The process of database validation is important and should primarily include an evaluation of the validity of the recorded data, an evaluation of the ability to complete the role for which it was developed. Critical to this process is the fact that data validation relies upon that the laboratories supplying samples should be reputably accredited, for example to ISO 17025. In addition, it is preferable if the laboratory is able to demonstrate participation in relevant proficiency testing schemes, and it is generally recommended that laboratories provide proficiency test performance metrics, e.g. Z-scores, prior to data being accepted for integration into the database. Database validation frequently relies on the use of resampling or cross-validation approaches. With this form of approach, a representative dataset is first assembled, and then the database compiled using a subset of training samples. Validation may then be performed using a further subset of samples reserved from the original dataset for the process. Alternatively, external validation may be employed. For this validation approach, the database is used to predict the validity of a set of unknown samples, which are not adulterated, have been chosen to be representative of inherent sources of variation, e.g. years, seasons, instruments, etc., but which is normally in excess of that seen in the samples used to generate the database. This approach is useful in determining the levels of false positives likely to be generated when employing the database. A consideration that can be overlooked is the presence of outliers in the data employed in constructing the database. During validation, the presence of outliers within the data may become apparent. Outliers are observations that are observed to be outside of the range of expected values and may need to be discarded. Standardized classification of outliers with databases for non-targeted analysis remains open to scientific discussion. they may be the result of analytical and random errors, or in some instances, as the result of a flaw in the assumed hypothesis. When outliers are present, the first action undertaken will be to confirm that they are not due to analytical or random errors. If following a reanalysis of the data, the samples are shown to differ from the original data, then initial results may be removed. However, where data are removed due to acquisition errors, consideration should be given to any samples analysed within the same batch. Outliers may frequently be attributed to particular and unique technological or geographical issues, such as a particular microclimate or a technological choice. In this case, further investigations are needed to understand if outliers belong to another population of data, or if they are just outliers falling in the percentage of error of the chosen confidence level, for example, 5% for 95% confidence level. In the case where an outlier is removed from a population within a database, for example, as a consequence of geographical issues, then the scope of the database may need to be reduced, and this should be noted so the database is not erroneously applied. Once a database has been created, appropriate curation is required in order to ensure that results remain valid over time. This is particularly important where the power of discrimination is not fully understood, and may be influenced by seasonality, changes in manufacturing process, replacement of analytical equipment, or software upgrades or changes. As part of this process, authentic and fraudulent samples generated following database constructions should be tested periodically to ensure that classification results remain consistent. In addition, to retain database utility, additional samples independently guaranteed as authentic may be employed to augment the database's content. However, challenge samples, even when shown authentic through database testing, should not be used to augment the database. Analysis of known authentic reference material, newly acquired alongside challenge samples, will help in ensuring that the database remains valid over time. Finally, where database sharing will be undertaken, special consideration needs to be given to who will be given access to the database, what levels of access will be awarded, for example, administrator, curator, or user. Although it is beneficial to share data in order to protect the value food chain, unlimited access may provide criminals with useful intelligence on how to avoid detection. Consequently, it is generally recommended that gatekeeper-style systems are employed where logistics permit. This permits the screening of potential user credentials and the allocation of appropriate access levels. Conventional practice has been for an organisation or project to generate their own resources or database specific to their local needs. The level of investment required in the development of a food authenticity database for use with non-targeted methods may further reduce the incentive for a developer to share data. The consequence is that there is a general lack of confidence in database validity for proprietary databases offered as a commercial service, primarily due to any relevant development process detail being retained on the basis of confidentiality. In addition, the difficulty of employing the results generated from the use of this analytical approach as evidence in a court of law is problematic, primarily because of the difficulty in explaining the underlying analytical concepts to non-experts. To summarise, non-targeted multi-analyte methods simultaneously analyse multiple analytes or markers and compare the resulting profiles to relevant reference datasets maintained in specialised databases. These databases typically relate to spectroscopic or spectrometric fingerprinting-based analyses. The application of databases for non-targeted multi-analyte analysis is a multiple-step process comprising of identification of authentic sample profiles or fingerprints, data processing to identify analytes which best differentiate between reference and other populations, construction of discriminatory models using the selected analytes, validation of database performance and records authenticity. Database validation generally includes evaluation of validity of the data recorded in the database, as well as the ability to complete the role for which the database was developed. Typical validation approaches may include resampling with authentic sets of training samples, or alternatively, an external validation using authentic non-training sample sets. Databases are comprised of underlying structural software, which organises and stores the data, and a user interface. Range of commercial and open source software available to assist with developing customised databases. Once developed and validated, database curation is necessary to ensure that results remain valid over time. Where database sharing is undertaken, it is recommended a gatekeeper system be employed to regulate user access. The following links and guidance documents may prove useful as sources of additional information. This e-seminar has been produced as part of the Joint Knowledge Transfer Framework for Foods Standards and Food Safety Analysis, and which is funded by the Department for Environment, Food and Rural Affairs, the Government Chemist Programme for 2020 to 2023, the Department for Business, Energy and Industrial Strategy, the Food Standards Agency and Food Standards Scotland.